have been having a great time with all the talks, and although I have not been able to catch them live, I was able to go through the YouTube channel and watch it, which I thought was really good. So thank you for that. What I'm going to be talking about is actually a family of algorithms that we're working on. But I'm going to focus just in the first one that we came up with, that is NANUQ. This is joint work with Elizabeth Allman and Joe Rhodes from the University of Alaska. And so, as mentioned in the title, sorry, we're interested in species networks. And when I'm talking about a species network, I am actually interested in this kind of object to the left, which each of these weird shapes represents a population. The length of that tube, or these weird tubes, represents number of generations. And the width of the tube at a specific generation means the population size. So as you can see, we can have really different populations and number of generations in each population and population sizes. Here we have these two red ones that depict this hybridization event, that these two merge together to create a new species. Now, I will also use this depiction of a species networks, which is, this is just getting the topology, and it's how people usually or are more commonly represented. But usually when you see this kind of depiction, you can think that there's more parameters on the underlying topology, on the underlying network. We also will have, for each hybridization event, we'll have gamma and 1 minus gamma. Per hybridization event, gamma can be different, and I'm going to explain later what do I mean by that. So yeah, so this is what we are interested in. We want to get some information about the network. So okay, so sorry about this. I don't know why now I have the old version of a talk. So this is other slides, but I think I can work with that. Or give me just 1 second. I'm sorry, just 1 second. Okay, now we are in the right presentation. Sorry, so let me go back 1 slide. So here we were. So to infer the species network, what we usually have access to is gene sequences or DNA sequences. And these sequences are believed to arise from a gene tree. For this talk, or for now, we just think we have access to these gene trees. There's many ways one can go from sequences to trees. I'm pretty sure you're very familiar with this. So for now, just think about we have this collection of gene trees. It is well known that gene trees does not have to depict the same evolutionary relationships. And this is a very well-known data set of the heliconius butterflies, where you can see in blue how these two species are more closely related, but for other genes, we have these ones. And it doesn't have to be as clear as that. And the reason for this gene tree incongruence, it could be due to hybridization or incomplete linear sorting, gene loss, and other processes, even like error from constructing your gene tree. So there's many reasons for having gene tree incongruence. So for this talk, we're going to focus on just hybridization and ILS for now. And incomplete linear sorting, or ILS, really informally it's just like this process on how a gene tree can happen or can be embedded in some way into the network. So you can have different gene trees that can happen in the species network. So what we're going to work with then is the network multispecies coalescent model, which is a stochastic process model describing gene tree generation that account for both ILS and hybridization. This was introduced by Kubako and Meng in 2009. And I'm going to give a really brief description of the coalescent model, a discrete version cartoon of it. So here we have our species network. You can see all these tubes represent populations. They have different colors for different populations. Each of these lines represents a generation, and each line has little dots, which represents individuals. So the number of dots, it's just the population size at a given generation. So what you do is you sample an individual per species at the bottom, and then at random, you're going to choose its ancestry from the previous generation. And then you keep doing that all the way. There will be some moments in time where you can have populations with multiple lineages, but this will be independent, and then they just do that. And if by chance two lineages select the same parent, then that will call a coalescent event. And then you keep going with one lineage. And you will keep doing that until all your lineages have coalesced and you have just one remaining lineage. Now, in terms of the hybridization event, here we have this hybridization event. This lineage has probability gamma of going that direction and probability 1 minus gamma of going the other direction. These events are independent. So in this particular case, this one went this way and this one the other, with probability gamma times 1 minus gamma. So this is, again, this is a continuous process, it's a Poisson process, and it can get a little bit tricky to work with because the support is fairly complicated, because you can have all these different gene trees that can be embedded in the network and different metrics for those trees. So it's a little bit, it can get a little bit complicated to work with the coalescent. But from here we get our gene tree. It looks like that. So yeah, so we are assuming that our data, our gene trees, are arising from the network multispecies coalescent model. So the question we want to answer is, given gene trees sampled from the network multispecies coalescent model on a network, and I am already, that should not be there, I'm sorry. The computer, I was gonna give it in another computer, but time, but yeah. We want to identify properties of the network. So we have a collection of gene trees. Given those gene trees sampled in the network multispecies coalescent model, what can you tell about the network? And that is the problem we want to attack. Okay, so now I'm gonna talk a little bit about some of the combinatorial properties or some more combinatorial aspects of networks that we need to address for this type of problem. The first is we're gonna be talking about unrooted semi-directed phylogenetic networks. So this is a rooted phylogenetic networks where all the edges are directed away from the root, and we have our hybrid edges. Now, when we root this network, what we do is we suppress the root and we remove all the directions of edges that are not hybrid, but keep the directions of the edges that are hybrid. So in some sense, here we still have some information about the root in some cases, but not quite everything. For example, we know that the root cannot be in this edge because that will make a contradiction with this hybridization event. So we know that the root has to be somewhere there. So you still get a little bit of information from this unrooting process. Now, so a quartet network in this context, or a quartet, will be the network obtained for taking all the paths from the root to a set of 4 taxa, let's say A, C, D, and E. You take all the paths from the root to that. And then you remove everything else and then unroot that network. You remove the root. And what you get is just a network on 4 taxa that is semi-directed. And the only things that are directed are the hybrid edges. So yeah. Now, because the family of networks is often too large to say something really strong mathematically, for now, we're gonna restrict ourselves to a family of networks level 1, where a network is level 1 if no pair of cycles in N share an edge. What do I mean by this? This network, we say this level 1 because you can see how this would call a cycle in the undirected sense. I know that by definition, networks are acyclic, but if we consider no direction in the cycles, this is on the edges, this is a cycle. There's no other hybrid node other than this in this cycle. The same for this one, there's just, in this cycle, there's just one hybrid node, and in this case, it's the same. But in this case, if we consider this cycle, then I have 2 hybrid nodes. So that is not longer level 1. Okay, so one advantage of this is that you can classify the type of cycles you will find in a level 1 quartet network. And these are just either you have 2 cycles, 3 cycles that look like these, or a 4 cycles. And you can have combinations of those, and not all of them can happen at the same time. There's some rules, but you can always classify the type of cycles in a level 1 network. And this will come handy later. So what we're going to do now is we're going to use the information from quartet networks to construct level 1 networks. But everything will be based on the information you get from the Coalescent model, from the sample of gene tree from the Coalescent model. So there's a lot of, there was some previous work and Posterior's work based on this construction of networks based in subnetworks. Now, there's many differences in these works and the one I'll be talking about, because there are some really good works on how you can construct level K network and some families of networks based on substructures, but it's not always clear how you can get those building blocks for data. And we think that is really important so you can get all the steps. So yeah, and there's some other works too that have the same kind of flavor, but some of them fail to address ILS, like incomplete linear sorting, or gene tree incongruence. I think of this list, the work that is the most related to ours, and actually our work was motivated by this work, is the one from Claudius Elise Lemos and Cécile Launay, which is a really good work. And actually, we work under the same setting, and we use the same approach, although the tools and techniques are pretty different. But we believe that our work and this work, they can really complement each other nicely, and they can be both used and complement each other. So that is pretty nice. And I'm going to explain a little bit on that later. But I mean, the literature on this type of works is growing, and it's pretty interesting. it's pretty nice how the community is getting a lot of things done. So, okay, so the approach we're gonna use is we're gonna work with quartet frequencies with concordance factors. But first, I'm gonna give the general idea. So suppose you have a sample of gene trees. So you have a collection of gene trees. Let's say we have these 5. Then we know that for each subset of 4 taxa, you have 3 different topologies, unrooted quartets, quartet trees, or quartet trees already. So these are trees. You have 3 different. The frequency of these is just what is the proportion of trees in your sample where you can embed this topology. So in these trees, I can embed this one in 3, so that is 3/5, and the same for the others. So this is the idea behind our approach. As mentioned, this is following Claudia and Cecile, and what we use is concordance factors, quartet concordance factors. So quartet concordance factors, it's just a triple of probabilities of G trees under the network multispecies Coalescent. And really briefly, what this means is you have a network, and then the first entry, it's just what is the probability that you can embed this quartet tree in a gene tree sample for that. Really informally speaking is suppose you have an infinite sample from here, this will be just the frequency of trees where you can embed this one, the frequency of these where you can embed this one, and the frequency where you can embed this one. That's really informal. So these triplet, it's numbers that are non-negative, and they actually have to add to 1, because in a given gene tree, you can embed just one of these. So that is a point in the probability simplex, which the probability simplex is just that region in R3 that has points that are non-negative entries that add to 1. So what we have here is just a map based on the Coalescent model, a map that takes a set of 4 species and gives you a point in the probability simplex. Okay, so with that, we now can go through the algorithm. No, sorry. Before that is why this is relevant is because using concordance factor, one can determine if the quartet network has a 4 cycle or not. You can show that if the map or if the concordance factor lies in any of these lines, then you do not have a 4 cycle. Then you you can have a 3 cycle, a 2 cycle, or no hybridization at all. Moreover, depending on in which line you will end up, you can know some of the information of the actual network or tree. You can know the underlying topology. So in this particular case, you can see that for all of these networks, C and A form a cherry. And because in all these, then your concordance factor will lie here. And if you had some other cherry like CAB, then you will be lying in another line. When you have a 4 cycle, then your concordance factor will lie in one of these points. Moreover, based on the order of how the taxa appear in the cycle, you can tell by in which region the concordance factor ended up. So for this one, you have A, B, C, D, and therefore your concordance factor will lie somewhere in here because you have A, B, C, D. Now, as you can see here, we have this line extension where these two intersect, but generically, you can show that for generic numeric parameters, the concordance factors of quartet network with a 4 cycle will lie in this region. So what this is telling us is that you can use your concordant factors to determine whether you have a force cycle or not, and a lot about the structure of that network. I just want to point out that you will not be able to determine which one is the hybrid node in the force cycle. If you change that for that and with adequate parameters, you can have the same point in the simplex. But you still get a lot of information. Okay, now we're can go to the algorithm. So the algorithm we developed is called NANUQ, which is a word for polar bear in Inupiat. So this algorithm is the purpose is to infer topological species networks. I don't know why that's a truth. So it stands for Network Inference Algorithm via neighbornet using quartet distance. The input is a collection of topological gene trees on a taxon set X and a hypothesis testing level alpha. And the output, if it comes from a level 1 network under the multispecies coalescent model, it's going to be the unrooted species network obtained after suppressing cycles of size less than 4 and direction of hybrid edges in 4 cycles. What I mean by this is the following. So suppose someone gives you a sample of gene trees that were taken from this network, and you feed those to NANUQ, then NANUQ will give you this back, which what is this? This is this network after you suppress the root. The 2 cycles you don't get, you are not able to distinguish. These here, they're supposed to be these 2 cycles, you lose them. Also cycles of size 3, this one you lose. And here you have a 4 cycle, 1, 2, 3, 4. You get the 4 cycle, but you don't know what is where the edge, which one is the hybrid. No. So this is what we do. So I'm going to explain now the steps of NANUQ. I'm not going to use this example. So what I did is I sample 1,000 gene trees using hybrid lambda from this network. And I'm going to go step by step and show you how we obtain this network, which this network is exactly what we expect to be, the unrooted version of this network after suppressing these 2 cycles and 3 cycles. And here there's a 4 cycle, and here we have it without knowing the direction of it. Okay. So the first step of NANUQ is for each subset of 4 taxa, determine the critical quartet count concordance factors from gene trees. And as mentioned, this will tell us whether we have a 4 cycle or not. So you take your gene trees, and then based on that, you look at the quartet frequencies per each of the topologies, quartet topologies, and then that will give you a point in the simplex. Now, because we're working with data, if your point lies in this line, then you know that you don't have hybridization, 4 cycles, sorry, and then you know some of the structure, and if you lie outside, you know that you have a 4 cycle, and you know a little bit about the ordering of the tags and the cycle. The problem, because we're working with data, is you are not going to be exact, so there's noise, so you might be a little bit to the left. So how you will treat that is, that's noise coming from a tree, or it's just a network that was pushed a little bit to look more like a tree. So for that, step 2 is to apply a statistical hypothesis test to just discount with level alpha to judge the evidence that the quartet species network displays a 4 cycle or not. This is work by Allman, Rhodes, and Mitchell, where they developed this test. And what we do here, you can see all the observed or the estimated or concordance factors from the data. And what we do, we have the null hypothesis that the quartet tree is tree-like. That means there's no force cycle. You apply that test, and based on that, you either reject, like these red ones, we reject the null hypothesis, or you fail to reject. So here what we do is all the ones that are blue, we think that they are trees, or they don't have 4 cycles, or if they are the red ones, we think they have 4 cycles, and we know that the lineage. Then what we do is we use this based on the results of the test. We're gonna construct a distance between taxa at the similarity matrix. And we do this by extending the ideas of roads. It's a work of roads where he defined a quartet distance based on trees. Now we just extend that same ideas to networks. And I'm gonna explain the distance, but more or less it's based on this function where you have these 2 species. And that will be 2 times the sum of a function phi that depends on your 2 species of a quartet, and you sum over all quartets displaying A and B, plus 2 times n. N is the number of taxa minus 4. So now this function phi, what it is it takes a quartet that has A, B, and 2 other taxa, and you're gonna have as output 0 if the quartet is a tree of the form A, B, C, D. That means A, B are a cherry in that tree. 1/2 if it has a 4 cycle, but A, B are adjacent in the cycle, and I'm gonna explain that, but that means that they're just in the cycle, they are next to each other, either up or down. And it will be 1 otherwise, which means you have a tree that separates A and B, or it's a 4 cycle, but they're like antipodal. And I'm gonna explain a little bit better that. So before going to the next slide where I explain a little bit more these cases, note that here, if in always for each quartet, you have a tree where A, B are a cherry, this is always accounting for 0, and then you just have to account for 2 times 2n minus 4. And anything else, when you have something else that is not like AB forming a cherry, that will add to the distance between them, making it further and further apart. So the main idea is to get the distance between 2 taxa. Then for every quartet separating this species, you have to use the rules of phi. Phi is just as follows. If you have, for example, a tree relating A, B, C, and D, then this tree will contribute, for example, will contribute 1 in a row to the distance between A and C and A and D because they are apart, or it contributes 0, for example, to the distance between A and B because they are a cherry. The case you have a network here, a 4 cycle, here, in this case, you have A is adjacent. I know adjacent may not be the right lingo to use, but in terms of the cycle, it's like the nodes are contiguous. But A is adjacent of D, so we think that contributes just 1/2 to the distance between A and B, and 1/2 to the distance between A and B and A and D, and it contributes 1 to the distance between A and C. And the 1/2 is a little bit arbitrary. Actually, it comes from reasoning. So in this case, we don't know which one is a hybrid node, right? But in any case, doesn't matter where you put the hybrid node. Some proportion of times you will have one edge, and some proportion of times you have the other. And in that proportion of times, suppose this is a hybrid node. If you remove that edge, then you will have AD and BC, AD will form a cherry and BC will form the other, because now this will be gone. And we think about that could be happening half of the time. And if you remove that, then you will have A and B contribute 0 because they will form a cherry, and then D and C. But in all these cases, A and C will always be separated. And in some of them, A and D will be together and some will be separate. So that is where the half comes from. So once you apply these for all your taxa, you get your dissimilarity matrix, which is a symmetric matrix with 0 on the diagonal, which denotes the rows denote taxa, columns denote taxa, and a specific entry just is the distance between the equivalent row and equivalent column taxa. So step 4 is to use NeighborNet to construct a circular split system from the quartet distance, and then use the circular algorithm from DRESS to obtain a split network. I'm not going to go to the details of NeighborNet or the circular algorithm, but it's just how this distance is constructed is in order to construct this split network that I'm going to explain a little bit better what is a split network. But yeah, what we use for now is a splits tree, which is software with NeighborNet and the circular algorithm are implemented. So what you do is you input that distance and you obtain a split network. Split networks, people usually refer to them as implicit networks, if I'm not mistaken, and they are a tool for visualizing incomplete and ambiguous phylogenetic signals. And these networks, usually you have to be really careful on what kind of conclusions, biological conclusions you want to draw from them. And usually what this represent is you have lines that are parallel and of the same length that all are part of the same split. For example, these orange ones just show that there's some contradict or incompatible signals between taxa 4, 3, 1 and the rest of these ones. Or for example, these three that are in green, these show how you have incompatibility signal between 7 and 8 and everything else. Now, in how our distance is constructed, we can actually have a biological interpretation for our network. And we will be able to draw conclusions of a splits network to get from the implicit to the explicit network. And this is done because when the hypotheses are being satisfied, you can interpret, you are expecting some type of split network out of NeighborNet for our splits tree. And what you expect is this kind of shapes that we denote as darts. And right now here are depicted pretty beautiful, but they don't have to be as beautiful, but they will satisfy these combinatorial properties. And we show that there's a one-to-one correspondence between the darts and the split network and the cycles in the species network. So we explain how you can, how you should expect, what type of object you should expect when your input comes from the different type of cycles. And they satisfy some rules and how the boundary will look like. And then, you can generalize that when you have more connected components around it. So with these, we have this set of rules that you can use to, given your splits network, that is the splits network we get out of splits tree, you can apply these rules. And first is you identify your dart. Here we have a 6 dart, sorry. It's like this one. It just doesn't look as pretty, but it is combinatorially, it's just the same object. And then you can identify what will be your hybrid node. Here we have a 5 dart that looks a little bit ugly, but you can see how this structure is. You have your middle diamond and then two on the side, like here, your middle diamond, 2 on the side. So you can identify what will be your hybrid node. And here we have a 4 dart, which 4 dart are actually just a 4 cycle, like, yeah. So for those ones, you cannot identify which one is the hybrid node, but you can know that they are there. So using our rules, you detect your darts, you identify which ones are the hybrid nodes, you remove everything that is inside the dart, you suppress degree 2 nodes, and then based on the position of your hybrid nodes, you put hybrid edges, and then you obtain your network. And this network is exactly what we wanted to obtain from NANUQ. So one advantage of NANUQ is that it's fast and can potentially detect violations of the model hypothesis. And we think this is really important because there is software or there's methods, better say, that it doesn't matter what you feed them, they will give you an answer. And that answer may not be that answer may not be, could be just coming from a bunch of noise. So it's important in that kind of aspect. One can show that NANUQ is statistically consistent. The running time of NANUQ is m times n to the 4th for m gene trees with n taxa each, which that makes it fast. I mean, the Bayesian approaches have a lot of trouble in converging for a few species and stuff like that. And yeah, so I mentioned how I think this could be really good in terms of complementary with something like SNAC, that is the method of Cecile and Claudia. And we think that NANUQ, because it's pretty fast, you can use it on your data and to see if you satisfy the hypothesis. And if you do, also you can see based on how many darts you get, how many hybridizations you have. And then based on that, you can then use something like SNAC to get, to SNAC, you have to provide the number of hybridizations, etc. And I will give you a network and you can just validate. Yeah. So you can find NANUQ in the R package, MSC quartets on the CRAN for anybody that wants to use it. Yeah, and if you have suggestions about things when you're using it, we welcome them. Yeah. Okay, so beyond level 1 networks. So taking these ideas beyond, like extend these ideas to broader families, there's 2 really big challenges into this. The first challenge is which properties of the family of the network are identifiable. And this is crucial because you don't want to have a method that will output something that maybe it's just wrong because things are not identifiable. So this is a huge challenge because networks are complicated and they can become really nasty really fast. And the second part is develop a statistical consistent fast algorithm to infer these properties for the network, which are, I mean, it's also a hard problem in terms of what can you get from the data? How you can get from the data to infer these properties. So anytime, like we try to focus on these 2 things to whenever we want to work on broader families. And we have some preliminary results for networks beyond level 1 via concordance factors. The first one I'm going to talk is about the trio, which suppose you have a network, any type of network, And then what you're going to look is the maximum 2 connected components. So here I just color code them. And the tree of blobs is just the tree you obtain after contracting them to a single node. So I have here denoted how you contract them. So for example, you have this thing. You have these tags that are all coming together, so they just get compressed to that. Then what you do is we have, in terms of the coalescent process, some identifiability results in what things you can get for the tree of blobs. And then we also have preliminary results on the algorithm. This is joint work also with John Elizabeth and Jonathan Mitchell. And the output we expect to have is this thing, that is the unrooted tree of blobs. And I was really happy last session that I really enjoyed the talk of Cecile about some of these problems, and I looked at their work on identifiability of local and global features, and they show how you can identifiability from average distances of tree of blobs. So that was really nice. So yeah, and we also have some preliminary results on a family of networks that for now we just call Banyan Networks. The word Banyan comes from these trees that are, I don't know if people have seen them, but these are really weird trees that have a lot of kind of branches coming together, so they look really network-like. That's where Banyan comes. And I'm not gonna defining them formally because this definition keeps changing as we move forward in this project. But we have some preliminary results in terms of identifiability and also on the algorithm aspect. For example, here we have a splits network that comes from something like NANUQ, from NANUQ. And here you can see how you have the underlying split structure, Dart structure. So, and in fact, if you apply some of the rules, if you generalize them, you can get back your network. So yeah, and again, I'm using the same ideas. We can also use this NANUQ approach in other contexts, and this is another work that another thing we're working on. So suppose you have the coalescent process plus a gene tree substitution model. So suppose you have your species network, and then you sample gene trees from that, and then you have a set of GTR parameters, and you sample one, and then you generate a sequence, and then you keep sampling pairs of these, and then you concatenate those sequences. Then one can map the triplet of sequences to the simplex using the LogDet distance. So if you have a sequence of 3 species, you can assign the point of the simplex defined by the LogDet distance between A and B, A and C, and B and C, and then normalize that. And if you are not familiar with the log distance, doesn't matter. It's just a really nice distance. But yeah, I mean, you can have this map. And similarly to the concordance factor case, we show that you can distinguish 4 cycles. So in this particular case for this work, we're working with rooted triplets, and we have the assumption that the network is ultrametric. So yeah, but under this context, you can show that if that map of sequences to the simplex, if you lie in this region, you will have a 4 cycle, and you can tell what tax is in the middle. And if you lie in any of these, you know that you don't have a 4 cycle, and you will be able to detect the the underlying topology of that. This is still on the works, pretty in the works, all of these. And oh, well, no, sorry. This part is the theoretical part we have finished, but the next one is what it's pretty in the works, that to develop this algorithm where you have the sequences, and then based on that, you can determine whether you have a 4-cycle or not, and the structure. And then with that, we can use construct a metric, and that metric in the same work of Rhodes that I mentioned before, he also has one for rooted triplets. So we use that, then just use a splits tree again, and under the same conditions, we get a dart also. And but now we can get the root with some little tweaks on there on how you construct your dissimilarity matrix. You can get a dart. This is a dart. I promise this is a dart, even though it doesn't look like. And then that dart has an interpretation. In this same context, in terms of identifiability, you also do not get cycles of size 2 or 3 cycles, but you get anything big. You get the root. So yeah, thank you very much.